You know, back in 2012, a single scientific paper landed that would change the course of biology as we know it. It took a hidden defense system from bacteria and turned it into, well, the most powerful gene editing tool humanity has ever had. All right, let's get right into how it all happened. I mean, really think about that. What if you could find a specific bit of genetic code, hit backspace to delete it, or even type in a new sequence, just like using find and replace on your computer? This isn't some far-off sci-fi concept anymore. It's the reality that was unlocked by the discovery we're about to explore. The possibilities are just mind-blowing, and it all started with a really simple observation in nature. So where did this incredible technology even come from? Well, the funny thing is, we didn't invent it. We just found it. Turns out, for billions of years, bacteria have been in a constant war with viruses, and to survive, they evolved this incredibly clever, super sophisticated weapon. And that weapon is called CRISPR. Yeah, I know, it's a bit of a mouthful, but you can basically think of it as a genetic most wanted list for bacteria. When a virus attacks, the bacteria grabs a little snippet of the virus's DNA and stores it. It's like taking a mugshot. Then it uses that mugshot to create these little guide molecules that can spot the virus and destroy it if it ever dares to show up again. By the time our story starts, scientists had already identified a couple of the main characters in this drama. First up, you have the Cas9 protein. This was the suspected assassin, the molecular scissors that would actually do the cutting of the viral DNA. And second, you have the CRISPR-RNA, or cRNA for short. This was the GPS, the guide that holds the address of the DNA target. But here's where the real detective story begins. When researchers took these parts and tried to get them to work in a test tube, nothing happened. It was a total dud. They had the assassin and they had the address, but something really important was missing. So the setup seemed pretty logical. You take the Cas9 protein, you add the guide crRNA that knows where to go, and you mix it all up with the target DNA. The expectation? They'd see that DNA get chopped up into little pieces. But this is what they got instead. This picture is called a gel, and it's a way for us to see DNA. And that bright, thick band you're looking at, that's the target DNA, sitting there completely whole, totally unharmed. Zilch. It was like the scissors were jammed. Or so it seemed. Now, a failure like this is what leads to the big breakthroughs. It forced the team to think. And based on some earlier research, they started to suspect there was a third player involved, another type of RNA called tracrRNA. They thought maybe, just maybe, this was the missing piece they needed to actually turn the system on. And now, look at this. This is the moment it all clicks. See this lane right here. This time, they added that tracrRNA into the mix. And bam, the big, thick band of target DNA is gone. Instead, you see these two smaller, fainter bands down below. That is the smoking gun, the unmistakable sign of a successful cut. TracrRNA wasn't just another piece, it was the activation key. Okay, awesome. So now they knew the three essential parts to make it work. But the next big question was, how? How does Cas9 actually cut the DNA? When they looked closer inside the protein, they found out it wasn't just a simple knife. It was more like a really sophisticated pair of molecular scissors. It turns out Cas9 has these two separate cutting parts. They're called nucleus domains, with the names HNH and RuvC. The idea was pretty simple, really. One blade cuts one strand of the DNA's double helix, and the other blade cuts the other strand. And this slide shows how they proved it, which was actually really clever. They went into the lab and they intentionally broke one of the blades. They mutated it so it couldn't cut anymore. And when they did that, Cas9 didn't slice all the way through the DNA. It just nicked it, cutting only one strand. That was the definitive proof that you need both of these domains working together to get that clean, final cut. All right, so the system works, and we know how the scissors snip. But what makes it so darn accurate? Well, as it turns out, Cas9 is a very picky eater. It doesn't just cut randomly, it has to follow a very strict set of rules. And the biggest rule of all is something called the PAM sequence. You see, Cas9 can't just land anywhere at once on the DNA. It has to find this short, specific little sequence, in this case NGG, that's located right next to the spot it's supposed to cut. You can think of it like a docking station for a spaceship. If that PAM isn't there, Cas9 just flies right on by. No cutting happens. And to make it even more precise, there's something called the seed region. This is the part of the guide RNA that grabs onto the DNA right next to that PAM docking station. And the match here has to be 100% perfect. It's like the final secret handshake that confirms to Cas9, yep, you're in exactly the right place, go ahead and cut. So at this stage, the team had a really solid understanding of the natural system from bacteria. It was amazing, for sure, but it was also a little clunky to use in a lab, needing 3 different parts. And this, right here, is where we see a pivot from pure scientific discovery into world-changing engineering. The researchers asked a question that was just beautifully simple. They knew the crRNA tells Cas9 where to go, and the tracrRNA tells it when to cut. So they thought, wait a minute, what if we could just link them together? Could we make one single, easy-to-program guide that does both jobs at the same time? And that is precisely what they did. They took the most important parts of the 2 natural RNAs and basically stitched them together with a simple little chemical loop. What came out the other side was this single, elegant, incredibly powerful molecule, a programmable, single-guide RNA. And here's the payoff. The proof is in the pudding, or in this case, the gel. This image shows without a doubt that their engineered single-guide RNA worked perfectly. It guided Cas9 to its target and made the cut just as well as the natural, more complicated system. In that very moment, a powerful new technology was born. And we have to be clear, this wasn't just some small tweak or a minor improvement. Fusing those 2 RNA molecules into one simple tool was the key that unlocked everything. It's what turned a fascinating bit of bacterial biology into a full-blown revolution for all of science. You know, even in their original paper, the authors had a sense of what they'd found. This quote is such a classic example of scientific understatement. Considerable potential. What they really meant was, we think we just handed the world the keys to the entire genome. So why was it such a revolution? Well, first, it's programmable. You want to target a different gene? You just change the guide's address. It's also super simple, just 2 parts, the protein and 1 little RNA guide. It's efficient, it's precise, and it's incredibly versatile. It works in pretty much anything, from plants to mice to us. It basically made high-level gene editing accessible to almost any lab on the planet. And that incredible discovery brings us here, to this huge, profound question. The work done in that 2012 paper gave us a tool with almost unimaginable power, the power to literally rewrite the code of life. And that leaves us with the ultimate question, not just for scientists, but for every single one of us. What on earth should we write?